Hello and welcome to the Laboratory Architecture and Reactivity of RNA at the Institute of Molecular and Cell Biology in Strasbourg. Growing well-diffracting crystals is a critical step in any crystallographic study. In this video, we will demonstrate the use of a new microfluidic tool to grow crystals of a biomolecule and to determine its 3D structure by in situ X-ray diffraction.
The microfluidic device, called ChipX, presents several advantages. It is designed to miniaturize and facilitate crystallization assays by the method of counter-diffusion. Its setup is easy and performed with standard lab material. It is also optimized for the direct characterization of crystals grown in the chip by several X-ray crystallography. ChipX eliminates crystal handling and cryocooling steps and preserves the intrinsic quality of the crystals, which are analyzed in situ and at room temperature.
ChipX has the format of a microscope slide, 7.5 by 2.5 centimeters. It contains 8 microfluidic channels used as crystallization chambers, with a section of 80 by 80 micrometers and a length of 4 centimeters. The inlet on the left-hand side allows the manual injection of the biomolecule solution, which will fill the 8 channels up to the reservoirs. Crystallization solutions are deposited in these reservoirs on the right-hand side. The crystallization is triggered by a phenomenon called counter-diffusion, when the crystallizing agent diffuses into the biomolecule solution and creates a concentration and a supersaturation gradient.